Okay, today we're presenting this cell cycle. Why do cells divide? First, to replace old, dead, or damaged cells. Second, to allow living things to grow. Why do cells have to divide? Through the process of mitosis, each cell gets a copy of all of the DNA, grouped with 46 chromosomes. Cells have to divide because they can't grow past their size limits. Cells are limited because they have to be able to transport food and oxygen to the parts inside. If a cell gets bigger, the outside won't be able to keep up with the inside, because the inside grows at a faster rate than the outside. In conclusion, this is why it is mandatory for cells to divide. Here's a picture of the cells dividing. As you can see, each cell is dividing somewhere. Next, why can't the cell grow infinitely big? As the cell gets bigger, the surface area to volume ratio decreases. When a cell is small, it has a large surface area to volume ratio. A large cell would have to have a small surface area to volume ratio. If a cell grows too big, not enough material would be able to cross the membrane fast enough. So my cell type will be animal. Remember, mitosis is slightly different between animal and plant cells. As you can see, this is the plant cell and this is the animal cell. They look very different as well. This is the pie chart of the percentage it takes for each phase to go through. Interphase. Here is my drawing of interphase. It has chromatin, the nuclear membrane, a cell membrane, a nucleolus, and centrosomes. Here's the picture of the interphase. Interphase has a series of 3 stages: the G1 phase, the S phase, and the G2 phase. G1 phase is where the cell grows and performs normal activities. S phase is where DNA is replicated. G2 phase is where the cell prepares for mitosis. After these 3 stages, interphase can move on to mitosis and cell division. Prophase, which is stage 1, has spindle fibers and centrioles, which is called the mitotic spindle. It has a nuclear envelope, which, as you can see, is breaking down, and chromosomes. Here's the picture. During prophase, the cell prepares for nuclear division by breaking down the nuclear envelope, which is why there is a dotted line. The chromosomes condense, the mitotic spindle is formed, and the centrioles move to opposite sides of the cell. Metaphase. Metaphase is stage 2. It also has centrioles. The chromosomes are now lined up, spindle fibers, and mitotic spindle. Here's the picture. Metaphase. In metaphase, the chromosomes line up in the middle and attach to the mitotic spindle fibers. Anaphase. Anaphase is stage 3. It also has centrioles, it has spindle fibers, and chromosomes. Here's the picture. Anaphase. In anaphase, the spindle fibers shorten, causing the sister chromatids to move to opposite poles of the cell. Telophase. Telophase is stage 4. As you can see, the spindle fibers are now withdrawing from each other. There is chromatin, cleavage furrow, and nuclear envelope. Here's the picture. As you can see, it is separating. In telophase, the nuclear envelope reforms. The chromosomes disperse into chromatin, and the spindle fibers dissolve. Cytokinesis. As you can see, it is now splitting into 2. They have chromatin, centrosomes, a nucleus, and a nuclear membrane. Here's the picture. As you can see, they are splitting in 2. Cytokinesis. Remember, cyto means cell and kinesis means movement. Cytokinesis is the splitting of one cell into two cells. This is when the daughter cell divides. And that is the end of my thing, if it will exit. Okay. Goodbye.